Good morning, ladies and gentlemen. Welcome everyone on another NeuroEYEscience seminar. Our today guest is Professor Roger Traub from IBM Watson Research Center and also Department of Neurology of Columbia University in New York. His speech today will be continuation of our last topic, which I mean gap junctions. And Professor Traub today will talk about gap junctions and very fast oscillations. Just as an introduction also, I would like to tell about our discussion rules. So there is a live chat option which you can use and ask questions during the speech. And also below the YouTube video, there is an email on which you can directly send your questions. So, dear Roger, just the screen is yours.
Okay, thank you, Adrian, for inviting me. This is a new experience, and so I hope it works technically. And as Adrian said, I'm going to talk about gap junctions and very fast oscillations and pyramidal neurons. There's a picture of where I used to work, but I'm locked out at the moment and I can't go there. So these are the questions which I'll try to address, at least some of them. So question one is, what is the actual evidence for gap junctions between pyramidal neurons? This being a rather controversial area. If they exist, by what means could gap junctions account for very fast brain oscillations, such as ripples? What are the implications for epilepsy and going into hyperspace for normal brain function? And I probably won't get to number 4, which is a contrast between the mechanisms that I'll propose and other types of network oscillations that involve gap junctions, such as occur in the inferior olive. So one of the motivations for studying this question comes from this classic paper by Yuri Buzaki and colleagues that was published in Science in 1992. So I presume everyone is aware of sharp wave ripples in the hippocampus. There was a large literature on them in the 1980s, and it was shown at that time that they seemed to occur because of a large excitatory synaptic input into pyramidal neurons in the hippocampus. And this occurred during states of awake immobility and slow wave sleep. So subsequently, with improvements in recording technology, Yuri Buzaki and others looked at the fine structure of these so-called sharp waves or physiological sharp waves. And some of the data is shown in the upper right corner of the figure. So at the bottom where it says 2, this is the sharp wave as it would look in classical recordings. And the new finding was that superimposed on this is a very fast field oscillation, in this case roughly 200 hertz, that is designated as or called a ripple. So sometime after that, Andreas Dragun and Nikolaus Meyer and later Dietmar Schmitz developed an in vitro experimental model of these sharp wave ripples. And you can see some of their data at the bottom right of this figure. So in A1, there's I'm sorry it doesn't show the time scale, but roughly 1 or 2 hertz in the local field potential, there are these transients. And if you look at the fine structure, you see a sharp wave and a superimposed ripple with a frequency that is approximately the same as what you would have in vivo. So the advantage of this preparation is that it allows a lot of experimental manipulations with drugs and in multiple intracellular recordings that are very difficult, if not impossible, to do in vivo. So this is one motivation. How is it that these fast oscillations can come about? And probably the most well-known idea about them is that, or hypothesis about them, is that pools of interneurons produce the ripple. And one of the reasons for believing that would be true is that if you record intracellularly from a pyramidal cell during the ripple, you would likely see ripple frequency inhibitory synaptic potentials. And I'm going to offer an alternative way of explaining that finding. So a bit of a digression now about gap junctions and axons, which is sort of the main topic of the talk. This may be the first, I'm not positive, the first demonstration of physiologically of electrical coupling between neurons. And it's from 1959, transmission at the giant motor synapses of the crayfish. And the preparation you see at the top, so that there are two intracellular electrodes in this giant axon, which I'm trying to show here. Oops. And then another two electrodes in another very large axon, which is coupled to the first one. And what Firstpan and Potter showed was that this the synaptic connection is an electrical synapse, not a chemical synapse, and it has very interesting properties, which you can see at the bottom. So if they were to evoke an action potential in the giant fiber, which you see here, that is transduced or causes an action potential in the coupled fiber at very short latency. So you see the time scale down at the bottom, 1 millisecond. So in a fraction of a millisecond, one action potential induces through electrical coupling another action potential. So it's worth thinking about this for a minute, because generally when people think about gap junctions in the nervous system, they're thinking about low-pass filtering. And this is how electrical coupling would work sub-threshold. If you don't have a large amount of electrogenesis, then because of the resistance and capacitance of the membranes, the gap junction would act like, act like a low-pass filter. But in fact, in this regime, that's not what's happening. It's not a low-pass filter. It's passing spikes from one cell to another. And this ought to be familiar for people who have studied the heart, because cardiac muscle cells connect to one another in exactly this way, or not exactly, but more or less this way. That is, there's electrical coupling from one cardiac myocyte to another, which allows a cardiac action potential to pass from cell to cell, which is a good thing, because without that, we would all be dead. So this is the type of electrical coupling that I'm going to be talking about, not the more familiar one of low-pass filtering. So that was electrical coupling in an invertebrate. The existence of gap junctions between axons was suggested somewhat later. In 1967, this is a figure from a paper by Mike Bennett and colleagues from that year, and it's from the brainstem of a fish. It's ultrastructure, obviously, and where you see an A, these are axons, and the arrow points to what appears to be a gap junction coupling axon to axon. So this is somewhat surprisingly to some people, apparently a rather general phenomenon in neurobiology, and the question is, where does it actually occur, and what is its significance, if any, in mammals? So just to remind you what a gap junction is, it's a localized collection of gap junction channels, which you see looking sideways here. So this is from electron crystallography So this would be sorry here, this is a gap junction channel. Where it says M, that's a membrane. Where it says E, that's extracellular space. And where it says M again, this is a membrane of a different cell. And the gap junction channel acts as a tunnel, which you can see in part B, that allows ions to flow and small molecules. Molecular weight depends on the type of protein, but typically of order 1,000 or so. So the channel is made of connexin proteins, which assemble, as you see in C, in hexamers here, and leaving a hole in the middle. So this is called a connexon. There's a connexon in each membrane, and they attach together through van der Waals and other non-covalent binding forces to form this kind of a tunnel. And then if you have an assemblage of these together, this would be called a gap junction. Now, different gap junctions have different numbers of channels. There seem to be some of them where there are very few, like of order 5 or even less channels. And then there are other kinds of gap junctions of order 10,000 gap junction channels. The conductance and properties of these channels are modifiable by modulators, such as acetylcholine and dopamine, and they're dependent on intracellular calcium, and their properties are also dependent on pH of either extracellular or intracellular pH, which turns out to be very important. And not only that, the way that the channel depends on pH depends on the protein. So some gap junction channels open or increase their conductance with intracellular acidification or extracellular alkalinization, and others behave in the opposite way. And this is also important. So the most common gap junction protein in our nervous system would be connexin 36, which tends to open with extracellular acidification. But there are other gap junction channels, such as formed by connexin 45, which behave in the opposite way, and they tend to open with extracellular alkalinization. So before I go further, here are some of my colleagues who've worked on this. This is Andreas Dragun, who's now professor in Heidelberg. Dietmar Schmitz is Einstein professor at the Charité in Berlin. And Rafa Gutierrez is in Mexico City. And Hanna Monnier is professor in Heidelberg. This is Eberhard Buhl, who actually died some years ago in 2003, and Miles Whittington, with whom I've worked most closely. This picture was taken in 2002. Now, so going back to ripples, the next step in this story was the discovery, accidentally by Andreas Dragun, that it was possible to dissociate sharp waves from ripples. So in particular, he found that you could see ripples without the sharp wave. I don't know about sharp waves without ripples, but you can get ripples without sharp waves. And he discovered this by accident, doing experiments in hippocampal slices and with a field electrode in stratum pyramidale in CA1 usually, and he saw this. So this is what looks like multi-unit recording in the shape of a ripple. This is about 200 hertz, and he found that these ripples, these in vitro ripples, or ripple-like, whatever you want to call them, would persist in calcium-free media that block synaptic transmission. So the control, what's happening in part A of this figure is demonstrating that the calcium-free media does block the field potentials associated with synaptic transmission. And you see in B that instead of removing the ripples, you actually have more of them. And this is reversible. So that suggests that this phenomenon, which by coincidence or not, is the same frequency as you see in an in vivo ripple, does not require synaptic transmission. I'm going to show you in a minute that this is not a single unit. So this is a collective phenomenon, a collective oscillation occurring somehow. So one thought that came up was that it might be produced by field effects, coupling nearby neurons. And this seemed to be, I've lost the cursor here. This seemed to be unlikely because the amplitude of these extracellular potentials is in the tens of microvolts. So that the transmembrane perturbations produced by these fields would be a small fraction of a millivolt and unlikely to produce this kind of synchrony. So Andreas went on to do some pharmacology. The other alternative was that this depended on gap junctions. And of course, it's hard to find a specific gap junction blocker that doesn't have unpleasant side effects. So he used a variety of different blockers. This is one of them. This is the general anesthetic halothane, and halothane reversibly would block this rippling phenomenon. He also used carbonoxolone and octanol, and he did pH manipulations. So tentatively, we're going to say that you can get ripples, not necessarily the same as in vivo ripples, but some kind of ripple produced by electrical coupling. Now, so the next slide addresses the question of whether this really is a collective phenomenon and not a single unit. And the evidence for that is in B of this figure. So in the upper trace are the field potentials, which are all more or less the same amplitude, but with a patch recording from a pyramidal neuron, the correlate of a field spike can either be a spikelet, this miniature action potential, or a full action potential. So that means that this field potential cannot be explained solely by the action potentials in this neuron. It has to be collective. In C, you see some more examples of the field correlating either with a spikelet or a full action potential, and sometimes an action potential without the field, also telling you that this is not a unit recording. So then we wondered, okay, so there are these spikelets, which look like what Spencer and Kendall described in 1961 in hippocampus, and I'll show one of their pictures later. Where could these come from? So I did some preliminary simulations of two model pyramidal neurons electrically coupled in their dendrites, and I forced one of the cells to fire by antidromically stimulating it. So if you antidromically stimulate it, sometimes you get spikes or bursts. You could also get spikelets when the antidromic axonal action potential blocked near the soma. But if you look at the coupled cell, you see a low pass filtered version of what you have here at the top. And this kind of a slow potential is something that Andreas did not observe. On the other hand, just for fun, and rather perversely, I put a gap junction, a model gap junction between the axons of two neurons, again stimulated one of them antidromically. And then in the other cell, you also got a mixture of spikelets and full action potentials, which looked quite a bit like what was recorded experimentally. So this is in 1998. This led to our hypothesis that the pyramidal neurons are coupled electrically through their axons, which I should say has gotten me laughed at a number of times, although not so much recently. This, now on the next slide, is another demonstration of in vitro rippling. This is not in low calcium. This is just with blockade of GABA A receptors. So just to recall, the in vivo ripple is associated with ripple frequency inhibitory synaptic potentials, presumably mediated by GABA A receptors. But in this case, you can block in vitro now. You can block GABA A receptors with the drug gabazine. The ripples don't occur spontaneously anymore, but they can be evoked with a puff of potassium chloride, and they occur at a nice ripple frequency and are not associated with intracellular iPSPs. And this all seems rather peculiar, and how does one put this together? So this is my former postdoc, Andrea Bibig. So in 2000, we suggested a model that would be consistent with the in vivo observations of ripple frequency iPSPs, and also with Andreas Dragun's observations that you can get ripple frequency oscillations without synaptic inhibition. So it's sort of a block diagram, and I'll get into the details in a bit, but let me go through it bit by bit. The first idea was that the ripple generation is where I have it wrote in red. The action is here. It's the collection of pyramidal cell axons in some population of pyramidal neurons interconnected by gap junctions. And this, we suggest, was the sort of oscillation generator at ripple frequency. So assume for the moment that that's true, the output of that circuit, I keep losing the cursor, the output of that circuit will synaptically drive interneurons, and fast spiking interneurons will then be able to fire at ripple frequency, as observed experimentally. So they're following, the interneurons would then be following the oscillation produced by this circuit, and then they in turn would induce ripple frequency iPSPs in the pyramidal cells. On the other hand, these axons are part of the same cells, and so the prediction was that the action potentials observed in pyramidal neurons during a ripple should be antidromic. And this has been shown in vitro. The in vivo situation is more complex, and I probably will not want to dwell on it, but the in vitro, this is the prediction which has been verified. So this is a bit complicated, so let me just concentrate on this part of the story. So the first prediction is, or the first bit of evidence we would like, is that you can get a spike in one neuron inducing a spike in another neuron. So I showed before that occurs in crayfish, and there's evidence that this can occur in pyramidal cells. So people object to this because it was done with sharp electrodes. I'm not sure that that's a valid objection, given that patch recordings also can perturb the intracellular medium of neurons and alter the properties of gap junctions. But in any case, here's the evidence as it was published. This is from the lab of Alex Thompson in England, and she was doing dual recordings between CA1 pyramidal neurons and also neocortical neurons. In this case, in D, she would induce a spike in one cell, the upper one, and that would produce a spike at short latency in the other neuron. Or you could get a spike inducing a spikelet shown in detail in C, looking very much like Andreas Dragun's recordings and the simulations in that Nature paper. So there's at least some electrophysiological evidence that this type of coupling exists. Another problem that people have with this story is that, oh yes, so the coupling exists, but it's actually very rare, and therefore it can't be important, or it's probably not important. And that argument is based on a logical fallacy, which I'm going to try to explain in detail. But just briefly, the logical fallacy is, as long as one cell can produce spikes in two other cells on average, that's enough to generate collective phenomena that involve the whole population. So a familiar example of this is an atomic bomb, where you have nuclei splitting and releasing energetic neutrons that then induce other fissions, and you get a chain reaction and an explosion when one neutron induces more than, when one fission induces more than one daughter fission. Then you can get exponential growth and an explosion. So the same mathematical principle applies here. If one spike can induce a spike not only in this single observed cell, but also in some other cell, that's enough to produce collective phenomena. Now, if you stop and think, one cell connecting to two others, that's very sparse coupling in a population that might be hundreds of thousands of neurons. So I hope I've been able to make the point that the sparseness of the coupling is not a logical argument, provided that the coupling is sufficiently strong to allow spike transduction. So here's another example of functional electrical coupling, or what seems to be electrical coupling between two neocortical pyramidal neurons. And this is in rat prefrontal cortex. And in this case, if you look in the upper right, the burst of action potentials was induced by a current injection. And in a second cell, you see the series of spikelets at very short latency and action potentials. And interestingly, when the pairs of cells were filled, which you see on the left, the axons of the cells sort of look like they are touching each other. Now, of course, this isn't ultrastructure, and there's no way to say from this picture that there's a gap junction at these sites of contact, but it's the kind of arrangement where there at least might be a gap junction. And I'll return to later on whether there actually is a gap junction at sites of apposition like this. Now this data, excuse me, this shows what's called dye coupling in the hippocampus. This is from a paper by Dietmar Schmitz et al. in Neuron almost 20 years ago. And this experiment was done by Sebastian Schuchmann, the second author. So what he did was, with confocal imaging, he injected a fluorescent dye into this cell. If you look in part A up here, and this cell he could show was a pyramidal neuron, because if you look in D1, it has spines on its dendrites. So this is a pyramidal neuron. And then he did confocal imaging sort of in time and space, and was able to make a movie. And in the movie, he could follow the movement of the dye. This is the movement of the dye from this neuron to this neuron. And then by going back and analyzing in detail, he could follow the dye along this process here and into this process here and into this other cell. And then if he looked at high power at these two processes, which are responsible for the coupling, they lack spines, and they have collaterals that come off at right angles here. So here's one coming off at right angles, and this one is really hard to see. But in any case, no spines, collaterals that look like this, which suggests that these are axons and not dendrites. So he was able to repeat this a number of times. He showed this kind of dye coupling in over 30 pairs of cells, and he was able to follow what appeared to be axon to axon in four cases. So one of the objections which was raised to this was that, how do you know that the dye is going intracellularly from cell to cell? Maybe it's extracellular. And I never understood this, because you just don't see any extracellular dye. But okay, that was an objection that was raised. Now, another piece of evidence, which I think is very beautiful, comes from immunofluorescence that was done by Jim Nagy, or Hungarian Nagy, but people call him Nagy, and he is in Manitoba. So he is a specialist, among other things, in developing antibodies to particular connexin proteins. And using an anti-connexin 45 antibody, he did these experiments. So this is hippocampus from rat in CA1, and he did dual labeling with ankyrin and the connexin 45. So the ankyrin is labeled in green up at the top here, and what it does is it allows you to see the proximal axons of these pyramidal neurons. And then the counter label is for connexin 45, and if you put them together, you see spots of connexin 45 labeling on what appear to be the proximal axons of the pyramidal cells, just where they were predicted to be from Sebastian Schuchmann's data and from simulations. And if you look at this in more detail, this is what you see. So there are spots of at least labels of connexin 45, not necessarily forming gap junctions. And interestingly, he was able to find spots at crossing sites. It would be wonderful if I could show you a freeze fracture to prove that there really is a gap junction there, but I can't do that. This is sort of the data that we have so far. And I ask the audience and my host and anyone who's might be listening to consider, if they're able to do freeze fracture, to look at these sites on the proximal axons of these pyramidal neurons. So these images are in a paper that was in reviews in Neurosciences, where I'm the first author, and it came out a few months ago. So correlated with that is this type of information that came from the lab of Nelson Spruston at Genelia Farm, and they looked at RNAs that were expressed in various hippocampal principal neurons. And when Nelson saw the at that time unpublished images, he said, "Oh yeah, we see the gene for connexin 45 expressed at high levels in ventral CA1 pyramidal cells." So this is also at least correlative, suggestive information that the protein might be expressed in those cells. As I say, we really need freeze fracture. So this is sort of historical interest. This is from 1961, I mentioned before. These experiments were done by Alden Spencer, who died many years ago, and the famous Eric Kandel, before he got into aplesia. And they were doing recordings in cats in vivo under anesthesia. And they discovered these, what they called fast prepotentials, which we now prefer to call spikelets, looking very much like what you can see in vitro. And I'm showing this partly because I just am fascinated by it, but also to demonstrate that the spikelet is not an artifact of cutting a slice. So this is some experiments, again, from the done by Dietmar Schmitz from the 2001 paper. These experiments were done in low calcium, blocking synaptic transmission, and he would evoke either direct antidromic action potentials, which you see here, or these funny looking action potentials that had a shoulder on them that looked exactly, to my mind, exactly like what Spencer and Kandel observed in vivo. So this arrow points to the shoulder. And you notice that this spike is coming off a hyperpolarization. So this is something that is not apparently not induced by synaptic excitation, but perhaps by antidromic coupling of some sort. So this is the kind of a schematic of what we proposed in that Schmitz et al. Paper, that there's a gap junction between the proximal axons of pyramidal cells. Dietmar's experiment, or most of the experiments, involved stimulating an action potential in one axon, and then he would record from a different cell. And the hypothesis was that there was a jump from here to here, and then antidromic propagation back here that would produce either a spikelet or an action potential, depending on membrane potential. And to test that hypothesis, he would block sodium channels here and block the spikelet, or he did multiple recordings of the axon and the soma, or the soma and the dendrite, and he showed that this spikelet potential was propagated backwards or antidromically into the second cell. And then finally, he showed that in this paradigm, with carbonoxylone, a gap junction blocker present, the spikelet, the amplitude of the spikelet was significantly reduced. So these experiments were all consistent with this schematic, although I would say they don't definitively prove it, but they're all consistent with this. So now if we accept the notion that maybe this kind of coupling does exist, which I show in this sort of diagram on the left here, how could it account for ripple oscillations in a network of cells? That still remains a mystery. So when people think of gap junctions and oscillations, the most common, most familiar model would be of oscillators that are coupled together by gap junctions. And if the intrinsic frequency of the oscillators are close enough, then it's possible to get a synchronized network oscillation. Although if they're not, the intrinsic frequencies are not the same, all kinds of other weird things can happen. So we don't think that that's what's happening here, because the coupling between cells is very strong, and because we don't have any evidence that these cells are acting like intrinsic ripple frequency oscillators. So ripple frequency oscillations are not seen in cells when they're uncoupled. So the problem is, if you were to couple cells together, how could you get a network oscillation? And the problem for a theory is, if you have a network oscillation, you have to have a sense, or not a sense, you have to have a description of where the frequency comes from. So this proposed a model for how that could happen, which is consistent with all the data that I'm aware of. And what's interesting about this model is that the frequency of the oscillation does not depend on a synaptic time constant. So unlike many other network oscillations, and we know that because they occur when chemical synapses are blocked. But in addition, the frequency doesn't depend on the time constant of a refractory current in the neurons, like a slope, like a potassium current. So what determines the frequency? And it's two things. The first is a few things. The first parameter is the time it takes for an action potential to go from this axon to this axon. And indirect evidence indicates that that is a fraction of a millisecond. So to go from here to here, fraction of a millisecond. So another parameter that determines the frequency is something called the mean path length in the network. So that is, if you have cells that are coupled to other cells, and you think of this as a graph, then there's a parameter that tells you, on average, how long does it take you to go from one cell to another cell, if both are picked randomly. So that's a topological property of the graph, and that is a main feature that determines the frequency of the oscillation. So this is something that's quite novel. It's pretty simple if you think about it, but it's also just different from what people are used to. So let me show some evidence that this occurs. This is at the bottom here. This is a simulation of thousands of cells coupled by their axons, demonstrating ripple behavior in a single cell. I'm not showing the mean, but the average potential will also be ripple-like. And you notice the interplay between full action potentials and spikelets. So how could that work? So this is trying to give some intuition. Again, this is a schematic of what we're talking about for a pair of cells with a crossing time, spike to spike, estimated to be a fraction of a millisecond. Then we put together a network of cells where each neuron is electrically coupled to more than one other, in this case, to two others. This doesn't show the whole network. It's sort of what you would see locally. Now, if you make a big network that's randomly connected with average connectivity that looks like this, and you have some low frequency of background spontaneous action potentials, what's going to happen? So this spaghetti here is a hand-drawn picture of a random graph where each number is a node in the graph and the lines are the connections. So of course, connected cells can't be too far away from each other, but when you try to draw a picture of it, you end up with these long lines. It's unavoidable. In any case, suppose you have a spontaneous or ectopic action potential at this cell where the red, and we've assumed that spikes can propagate. So in this case, it propagates to a few other cells, the blue ones, and then to more cells, the green ones, and so on. Eventually, it fills up the whole network, and you have collisions, and because of refractoriness, it dies. So that will give you a wave of activity. And the time it takes to go from this event, the red event, to the peak of the wave, you can see is going to depend how long it takes to go from one node to another, which is this time, and how long it takes, how many steps on average it takes to go from here to some randomly chosen other cell. So intuitively, that's where the period of the oscillation comes from. Now, it's not clear from this that you would get a sequence of waves, and I don't have a good theory to convince you of that, but I do have lots and lots of simulations that show that if you have random Poisson-like noise in this system, it produces waves that have, it's not a delta function Fourier spectrum, but a narrow Fourier spectrum. And it looks, in other words, it looks like an oscillation. So that's the theory that, that we've advanced. Now, just as a bit of a digression, there are other types of gap junctions in the mammalian nervous system. These are called mixed synapses, and Jim Nagy and John Rash and others, and many others, have looked at the anatomy of these quite extensively. But Rafa has also looked at the physiology. And this came about in part by a chance discovery by Farid Hamzai-Sichani, who was a former student of mine. And he was filling CA3 pyramidal cells with a fluorescent dye and making these beautiful confocal images. And from time to time, he saw the dye after an injection filling processes like this that are not dendrites. They lack spines. They're coming off the proximal dendrites of the pyramidal neuron, and they look actually like mossy fibers. And that suggested that there was a gap junction between the dendrite of this pyramidal cell and the mossy fiber, which would imply that the classical mossy fiber pyramidal cell synapse is a mixed synapse. In other words, it has a chemical component and an electrical component. And you can actually see ultrastructure ultrastructural evidence for that. This is at the synapse. This is a chemical synapse. These are vesicles, and there's a gap junction. And these have been immunolabeled. So these particular gap junctions are connexin 36, not connexin 45. So Rafa has, this paper of Vivar et al. Looks at the physiology that you would expect at this type of mixed synapse. You can see here the electrical component, a spikelet, followed by a more classical EPSP. Something very similar to what you see in the Mautner cell in goldfish. And in any case, Rafa has a recent paper in Journal of Neuroscience that just came out showing that these mixed synapses are extremely common, but mostly they're latent or silent. They're only physiologically visible under conditions of appropriate intracellular pH. That's a digression, but something very interesting I wanted to mention. Now this picture is to just make the point that long time constants can come about even though the crossing time cell to cell is very short. So that's what I propose is happening in this network. The crossing time is, say, 1/4 of a millisecond, but the oscillation period is 5 milliseconds. So 20 times slower. And that's because to get a wave of activity to go from this cell to sort of the whole system takes of order 20 steps, or depending on the size of the network, but many more than one step. So the same phenomenon is actually familiar to anybody who's studied electrocardiograms. So the period from one QRS complex to another one, which you don't see here, is this is determined by intrinsic oscillatory properties of the pacemaker and the sinoatrial node. But the time, the PR interval, the time to go from the P wave to the R wave depends on conduction cell to cell to cell to cell within the heart. So the action potential cell to cell is rather short, but because there are so many steps, you can have a PR interval that is quite long relative to that time. Now, so in the last minutes, I want to say something about the ripple frequency oscillations that occur prior to seizures in at least some patients with focal epilepsies. So I should have included, maybe I can close this. So this is from a paper in Epilepsia in 2001. And this, so I was first author. This was done with Miles Whittington and with child neurologists at in London at the Great Ormond Street Hospital for children. And they looked at a series of patients, all children with focal cortical dysplasia, so-called. So it's a region of improper development of the cerebral cortex. And these children all had intractable epilepsy, and in each case, with appropriate workup, the epilepsy began in the vicinity of the dysplasia. So the kids were investigated with a so-called ECOG or electrocorticography. So this is invasive recording with multiple electrodes on subdural, so-called subdural electrodes. And this is an example of the onset of a seizure in one of the children. So these very big, these high amplitude waves, this is what's called the electrographic seizure that you see in many, or actually all of the recording electrodes. But what Torsten Baldeweg and the other child electroencephalographers noticed was that in every seizure, in every child, the onset began with rippling, which you can see at low amplitude here. This is at roughly, in these children, roughly 100 hertz, and it would go on for a period of seconds and then turn into a seizure. And we wondered if the physiology of underlying this abnormal, presumably abnormal focal rippling in the cortex might have the same principles as the normal ripples. So this is some experiments that I want to show you in vitro and also human data from a later paper in 2010. So at the top is, again, ECOG, electrocorticography, this time from an adult patient. So this is 11 seconds of recording. This stuff here is the electrographic seizure, which has been cut off. But in this patient, every seizure also began with something that looks like a ripple, which you see down below. And its power spectrum shows a peak at roughly 150 hertz. Again, rather like the children with the cortical dysplasias. So this is, to me, a very striking experiment that Miles Whittington did. He was able to replicate that phenomenon in vitro. This is in a rat slice where the chemical, he used a cocktail to block chemical synaptic transmission. And if he acutely alkalinized the tissue, which would open connexin 45 gap junctions, he got a field ripple, which you can see in B, this. And he did simultaneous recording from a large layer 5 pyramidal neuron, the so-called IB, or intrinsic bursting neuron. Showed again this, we've seen many times before, this runs of spikelets and action potentials mixed together, and the action potentials are in phase with the field ripple, like so. Very similar to the human data, although of course we don't have intracellular recording. Now, this is an expanded view of the experiment with the field and the cell. And this is from a simulation, again, from thousands of model pyramidal neurons electrically coupled through their axons, such that spikes go from one to another, such that each cell is coupled to more than one other cell. And it looks very, 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 very similar. So the prediction then is that the onset of seizures may be associated with conditions which produce this kind of rippling behavior, and at least experimentally, that can be caused by a change in the pH of the brain. So this opens up an experimental avenue for epilepsy, and the question is, at the onset of a focal seizure, is something happening in the tissue, in the brain tissue, that changes the pH, that opens these gap junctions and starts the rippling. Now, that's not going to explain the seizure itself, but it might explain the onset of the seizure, and which would produce conditions that are then favorable for the seizure proper. And if this is correct, and we don't know if it's correct, but if it's correct, then that would be a new target for a possible treatment, namely one that's aimed not at the seizure, but aimed at whatever is happening just prior to the seizure. So I think it's time. Oh, one last slide. This is the prediction. I mentioned that spikes were antidromic in our models of ripples. So this is a simulation of a sharp wave ripple showing hyperpolarization and then an action potential. So this is not a synaptically induced spike, but an antidromic one. And this is an example of an experiment. It doesn't prove that the spike is antidromic, but there's indirect evidence, and there is very direct evidence that it's coming off a hyperpolarization, and it looks like an antidromic spike. Now, that doesn't mean that spikes during in vivo ripples look exactly like this, and there's some evidence that they don't, but it would be interesting to do further tests, and some have been published already on whether the in vivo spikes are antidromic or not. So that's, yeah, I don't want to get into too much detail. This is a somewhat controversial question about what's happening in vivo. Yeah, so thanks to supporting institutions and many colleagues who have participated in this, especially Miles Whittington and colleagues in Germany, such as Andreas Dragun, Nikolaus Meyer, and Dietmar Schmitz, and of course, many others. So thank you. Oh, this is a plug for our book.
Thank you, Roger, very much for this really interesting and detailed talk. There's one question which I would like to ask you. Do you think that the fact that gap junctions are coupling proximal neurons, may it be the way for neurons to kind of create a system of filtering or cutting off some of the impulses, like for example in case of visual transmission? Is it possible, do you think that it may have function on kind of buffering or cutting off or filtering some of the signals?
I don't know. The way I would think about it, so you're talking about your work on the optic nerve?
Yes.
I'm just going to make a guess that what's involved there is a gain control. Yeah. That under conditions when the gap junctions are closed, you're looking at fine details, and this is what you would want to do in light conditions. And in the dark, you can't see the fine details, but you want to pick up anything that you can. And in that case, there might be an advantage to having pools of axons that are coupled together just to amplify the signal. Of course, you don't want to amplify it too much, or I guess it would just be a big flash, which is not helpful.
Yeah,
that would be my guess as to the significance of what you were observing.
Yeah, I just asked because most of people are talking about gap junctions more from this let's say chemical side, that it allows transfer some neuroprotectants, or it can participate in spreading the apoptotic signals, but yeah, it's quite seldom people talk about this real electrical function. So right. In my opinion, this amplification aspect is really fascinating.
Yes, and it could be really critical.
Yeah, and could
for our ability to see in the dark.
Yeah. Okay. So once more, I will remind you that there is an email address, so you can directly contact Roger and ask your questions if you have any. And from our side, we really thank you as a NeuroEYEscience for you to join our seminar series and giving this excellent talk. And I hope that there will be more in future. And thank you very much.